morning, everyone. Today I am going to take Pinus Reprodction. In continuation of yesterday's class, today's topic is Pinus Reproduction. Pinus reproduce sexually by means of formation of cones. Cones are nothing but strobili, which are equal to the flowers also. In angiosperms, reproductive parts are flowers. In gymnosperms, instead of flowers, cones are produced to do the reproduction. So coming to the sexual reproduction of Pinus. Pinus is a monoecious sporophyte. Monoecious means nothing but on single plant, different types of cones are produced. It means male and female cones are produced on the separate branches on the same plant. It is known as monoecious. Sporophyte means the plant which produce spore-bearing organs on it. Spore-bearing organs nothing but they are cones itself. So, Pinus is a monoecious sporophyte. Monoecious and dioecious, there are two types of plants we can find out in dicotyledons as well as in monocots, also in gymnosperms. Coming to Pinus, it is a monoecious and netum. One more advanced gymnosperm is a netum, that is dioecious. It is the equal similar quality of dicotyledons. Dioecious means production of male and female reproductive organs on separate plant. Plant is separate, male plant, female plant is separate. Such a condition is called dioecious. But whereas monoecious is the primitive character, on the same plant, on the single plant only, male cones and female cones are produced on separate branches. Now coming to the first. The male cones are developed on the lower branches, while the female cones are formed on the upper branches. The male cones which replace the dwarf shoots. Look at the picture, we can find out these are all male cones. In a cluster they are formed. This is the spike axis. These are all the needles. These male cones are produced or they replace the dwarf shoots. They are produced or developed in a clusters on the base of the current year's long shoot at early spring. So coming to the first one, first the reproductive organ which is known as male cone. Male cones are not in a single. Male cones are produced in a cluster. They are produced in the replace of dwarf shoots, and early spring they are produced in more in number. And the number of male cones are also vary from one species to another species. If we see some examples in Pinus Walchiana, the number of male cones are 15 in number, 15. Pinus Walchiana lo 15 male cones are. And in Pinus Roxburghi, there is formation of 140 male cones. So here the number will be differ. The formation of male cones, so minimum to 15 and maximum to 140. So remember this point. In Pinus Walchiana, they are 15 in number, male cones in a cluster. In Pinus Roxburghi, they are 140 in number. And at the onset of spring, the male cones fall off and simultaneously the young female cones are born in pairs in a cluster around the tip of the long shoots. The number varies okay. Now coming to the male cones. So if we see the male cone structure, it is a stout and compact aggregate structure. Look at this. This is a single male cone. Like that, several male cones which are formed in a cluster. So it is a stout and compact, which is formed or made up of by so many microsporophytes. Each male cone is very small in young stage. Approximately the length is 2 to 4 centimeter and the shape of the male cone is in oval shape, and male cones are naturally developed in the axil of scale leaves. Each male cone is consisting of a central axis. The central axis which is spirally arranged by 60 to 150 microsporophylls. So each male cone consists of minimum number of microsporophylls are 60 and the maximum number of microsporophylls are 150. Like that, each male cone is a compound, compact, tight, brown color structure which is made up of with microsporophylls, and the microsporophylls are coiled or spirally arranged on the central axis and they form the male cone. Now coming to a single microsporophyll. This is nothing but equal to the stamen. A single microsporophyll is a triangular in shape. It is a membranous stalked structure with a distal expanded sterile part called apophysis. Microsporophyll is nothing but equal to the stamen. It is a triangular in shape and it is having a distal expanded sterile part is called apophysis. Each microsporophyll consists of two sac-like structures which are known as microsporangia on the lower side or on the abaxial surface. So, like in dicotyledons, how the stamen consists of two anther lobes, and those anther lobes are produced for the pollen grains or microspores. In the same manner, each microsporophyll consists of two sacs or microsporangia. They are equal to the anther lobes of dicotyledons, and these sac-like microsporangia are present on the lower surface of each microsporophyll. And here, coming to the development of microsporangia, it is eusporangiate type. It means that it develops from a group of cells, hypodermal cells of the microsporophyll. There is two types of development takes place in the microsporangia. One is eusporangiate and another one is the leptosporangiate. The eusporangiate means the structure which develops from a group of cells, that is eusporangiate. Leptosporangiate means the structure which is developed from single initial hypodermal cell. It is called leptosporangiate manner. So again I am repeating, microsporophylls are nothing but the triangular leaf-like structures which are present on the male cone. And these are equal to the stamens. Each microsporophyll consists of two sac-like structures, and each sac is produced for the responsibility of microspores. These two sac-like microsporangia present on the lower surface of the microsporophyll, and the development of microsporangia is eusporangiate manner. Now coming to the microsporangia, when it comes to the maturity, it splits longitudinally. If we take LS of or TS of microsporangia, it shows tetralocular, and that each locule consists of multi-layer epidermal cells and tapetum, and inner to the microsporangia there is a huge number of microspore mother cells are present. So here, if we see the structure of microsporangium consists of many layers. Wall layers are many, and tapetum is one important layer which is present beneath the epidermis, which is a nutritive layer, and that supply nourishment for the development of microspore mother cells. These mother cells undergo reduction division, and each mother cell, microspore mother cell, give tetrad microspores. That is the very common most character of among all the flowering plants as well as in gymnosperms also. So each microspore mother cell by meiotic division produce 4 microspores are nothing but pollen grains. Thus, at maturity, a single microsporangium contains numerous pollen grains. These pollen grains are pale yellow in color. And here the pollen grains are boat shaped with monosulcate aperture. Monosulcate means it is a small bracket-like board aperture present on the surface of the pollen grain, which is bounded by two wall layers. The outer wall is very thick. It is known as exine, and the inner wall is thin. It is known as intine. The exine on the lateral side expanded to form two wings. Here, coming to the important character of the pollen grain is, there is two sac-like balloon structures arise lateral sides by the expansion of exine. The outer wall of the pollen grain is known as winged pollen grain. Thus, the pollen grains of Pinus are indicating anemophilous mode of pollination. Anemophilous is nothing but wind pollination. So, wind pollination is naturally occur in monocotyledons in grasses and where the pollen grains are very light in weight. So, here coming to already the Pinus pollen grains shows adaptation for the wind pollination, means they consisting of two wings. Wings are for the fly. So, anemophilous is the very common method of pollination which is occur in the Pinus. And the breakage of microsporangia is a longitudinal dehiscence. It splits longitudinally, means in the straight manner. Now, coming to this, these are the pictures of Pinus male cone. Look at the first diagram. These are clusters of male cones which are produced in the axillary faces of scale leaves, which are replaced the dwarf shoots, and this is the pollen grain. Look at this, two sac-like wings are clearly observed here. These two sac-like wings are expanded from the exine, and this is a single microsporophyll. This is the sterile part is known as apophysis, and this and this two are theca or microsporangia. Now coming to this central axis, it is LS of male cone. It is the central axis of the male cone. There are 60 to 150 approximately microsporophylls are arranged spirally. Look at this picture. This is the one triangular microsporophyll, 1, 2, 3, like 60 to minimum and 150. And these are the microsporangia. In the center of the microsporangia, there are several microspores are developed by the meiotic division or by the reduction division of microspore mother cell. So again I am repeating, it is a less of female core, it is the central axis. On the central axis, spirally present a number of microsporophylls, and each microsporophyll is having two microsporangia, two theca on the lower side. In this view, we are finding only one. In 3D view, we can find out two microsporangia. Inside the microsporangia, these type of microspores are present. It is nothing but pollen grain. It is the winged one. It leads to the anemophily. It is a very common and important character of the Pinus. So, in pollen grains, which are released in large quantity and into the air, so that wherever the pine trees are present, the entire environment or the atmosphere is become into yellow at the time of pollination. So this is popularly called as sulfur shower, which occurs especially in the spring season. If we see the nature or environment during the spring, we can find out the yellow dust in the sky. That is due to the releasing of pollen grains from the pine trees. So this is clear cut about the male cone. So again I am repeating, in Pinus, male cones are the male reproductive organs. Male cones consist of microsporophylls. Microsporophylls consist of microsporangia. Microsporangia produces several microspores after the reduction division of the microspore mother cells. Those microspores later on developed into male gametophyte. So this is the clear cut about the male. So how the microspore developed into male gametophyte? That is the important thing. It is the gametophytic generation. Till now we are discussed about the sporophytic generation. Now we are coming to the gametophytic generation or the microspore. Microspore is the initial cell for the development of male gametophyte. So development of male gametophyte, look at this is the pollen grain, winged one, exine, intine, these are the two wings. And this first, the haploid nucleus which is present in the pollen grain, that will be divided unequally to produce one large cell and one small cell. The small cell is called prothallel cell and the large cell is called antheridial cell. And again the prothallel cell will be slowly disintegrate and one more division takes place in the antheridial cell and which is leads to the formation of second prothallel cell. Like that, first and second two prothallel cells are slowly degenerate. Now this antheridial cell will be divided into two cells. One is the generating cell and the other one is tube cell. And this tube cell will be expanded into tube-like structure, and this is the pollen tube which carries tube nucleus. And leftover generating cell will be divided into two cells, stalk cell and body cell. And this body cell gives us two male gametes. So like that, the single pollen grain, the haploid nucleus of the pollen grain, which gives us two male gametes. This is the entire big structure is called male gametophyte. Male gametophyte is nothing but the structure, the organ which consists of gametes. The structure, the organ which having haploid gametes, that structure is called gametophyte. This gametophyte is known as male because of it is developed from single haploid microspore. This is the initial cell for the development of male gametophyte. Again, I am repeating, male gametophyte is formed by the reduction division or by the meiotic division of the microspore mother cell. This mother cell, after reduction division, the haploid nature is attained by the microspore nucleus. This haploid nucleus divides once for the formation of one small prothallel cell and one large cell. The prothallel cell will be left over the first one. This large cell become into again division. These divisions are normal mitotic divisions, and this prothallel cell second one is formed and antheridial cell are formed. This antheridial cell divides once and formation of generative cell and tube cell. And tube cell is nothing but all the tube. This tube cell is also having one haploid nucleus that will be passed to the enter into the pollen tube, and generative cell will be divided into stalk cell and body cell. Body cell is nothing but which is responsible for the production of two male gametes. So here one microspore gives two male gametes. So this is the development of male gametophyte. Now we got the development gametes. Now these gametes reach to the ovule, that is known as pollination. So here one thing we have to remember, the pollination will be taken in the early stage at the time of two-celled pollen grain, or sometimes after development of male gametophyte also the pollination occurs. That will be depend upon the environmental situation. It means three-celled pollen grains are reached to the stigma, or this gametophyte also reaches to the stigma. That will be differ from one species to another species according to the generation. So here pollen grain, again this is the same thing. Pollen grain has two prothallel cells and an antheridial cell. These cells soon disintegrate. The antheridial cell get cut off and second prothallel cell is formed, and it also cut off generative cell, and the remaining large cell is called tube cell, and tube cell is left over like that. Body cell gives us two male gametes. Here, tube nucleus controls the growth of the pollen tube. Exine of the pollen grain ruptures, intine grows out to form pollen tube. Antheridial cell divides into two, stalk cell and body cell gives us two male gametes. These two male gametes, along with the tube nucleus, pass through the pollen tube and reach to its tip. Now, so that is exclusively complete development of male reproductive structure. So, male cone, microsporophyll, microsporangium, pollen grains. From the pollen grains, we will get two male gametes. So, till the microspore mother cell, it is the sporophytic generation. And the next, when the microspore is formed by the reduction division of the microspore mother cell, that leads to the gametophytic generation. So, formation of two gametes from single microspore is called male gametophytic development. Now, coming to here, the next second reproductive part is called female cones. The female cones, which are beautiful and which consist of naked ovules, and the female cones are nothing but which are produced in pairs. At the onset of spring, the male cones fall off, and simultaneously the young female cones are born in pairs or in cluster around the tip of the long shoot. Here, cluster means not like a male cones. Either they are one or single or solitary or maximum 2 to 3 in number. So, female cones are produced in pairs or in cluster in the axil of the scale leaf. The female cones mature very slowly and the growth may persist for several years. So, the female cones are, young female cone is very small and the size of the young female cone is 1 to 2 centimeter in length, and it is very soft and compact and red-green in color. The second year female cone is comparatively large. The size of the second female cone is 5 to 8 centimeters in length and woody, compact and green in color. The fully matured third year female cone is very big and stout, much larger. Approximately the length is 15 to 60 centimeters and it is woody and loose. The megasporophylls become into loose with the growth, with the expansion, and it become into brown in color. Each young female cone consists of central axis and it bears spirally arranged scales. Now coming to again female cone. Here the female cones are produced in the axils of scale leaves. Here the first year female cone is young and very small. The size is very small, 1 to 2 centimeters, and soft, compact and red-green in color. The second year female cone is comparatively large, 5 to 8 centimeters in length, woody, compact and green in color. The fully matured third year cone is much larger, 15 to 60 centimeters in length, woody, loose and brown in color. Like that, each after completion of the third year female cone, now it starts for the development and it is ready for the fertilization process. Now coming to like that, each female cone consists of two types of scaly leaves. The first one is bract scales, which are very thin and membranous and directly attached to the central axis. And the second type of leaves are nothing but ovuliferous scales, which are woody, present on the central axis surface on each bract scale. Each ovuliferous scale bears 2 ovules. It is nothing but megasporophyll. They are situated in the, see, look at this, side by side on the upper side. Now this is the female cone. So female strobilus, very clearly with the imaginary picture, ovule, seed, bract scale and central axis and woody, ovuliferous scale. This is the bract scale, ovules. this is the complete wood female cone strobulus, which is matured, which is ready for the pollination. This is the central axis. These are elongated triangular megasporophylls. Each megasporophyll made up of with two types of scales. One is the brachycell, which is very thin and membranous, and which is large ovuliferous scale, which bears two ovules on its lower or abaxial surface. So you can clearly see this ovules. These are modified seeds after fertilization. This is the first picture is the before fertilization. The second picture is the after fertilization. So ovules only naturally modified into seeds after the fertilization. Now coming to the structure. The ovules, which are two in number, present on the abaxial lower surface of the ovuliferous scales. Look at this. This is the type of ovule which is anatropous. And before start the structure of ovule, the female cone consists of number of megasporophylls on the central axis, which are spirally present. Approximately 80 to 90 number megasporophylls are arranged on the central axis. The brachyscales and ovuliferous scales thus form a seed scale complex. A single megasporophyll consists of two types of scales, that is over already, brachyscale and ovuliferous scales. And each megasporophyll is triangular in shape and which is having in the same manner a thick sterile part which is known as apophysis, and the development of megasporangium or ovule is used sporangiate manner, the same like superficial cells of ovuliferous scales. A group of number of cells are involved for the development of the ovule or megasporophyll. Now coming to the structure of ovule. The ovule is anatropous. There are several types of ovules as we have seen in the dicots. So orthotropous, anatropous, semi-anatropous, amphitropous, carcinotropous, and campylotropous. Like that, majority of the plants exhibit anatropous. Anatropous is nothing but curved ovule. Look at the picture here. This is the micropyle. The opening pore of the ovule is called a micropyle. And this is the basal part is called chalaza end. And these are the integuments. Here, unitectic ovules are single-coated, single-layered balls. So unitectic. This single jacket of the ovule is having inner fleshy, outer stony, outer fleshy and inner fleshy and middle stony layer. Single technique, the single jacket ball layer is called unitechnic ovules, and this single unitechnic ovule is having three layers. Outer and the innermost ones are fleshy and the middle one is the stone. Now, the centermost part of the ovule is occupied with the nucellus tissue. This nucellus tissue is diploid in nature. Look at this picture. This is the entire structure is known as female gametophyte. Now, ovule is a diploid sporophytic in nature. When the nucellus tissue, among one nucellus tissue, one cell which is present towards the micropylar side, which is act as a megaspore mother cell. Look at this. This is the micropyle opening pore for the entry of the pollen tube, and there is small pollen tubes are also visible in the ovule. There is some space where the pollen chambers, pollen grains are dropped and take some rest, and this is the entire tissue is the nucellus tissue. In this nucellus tissue, one of the cells which is present towards the micropyle side, which act as megaspore mother cell. That megaspore mother cell undergo reduction division and produce 4 megaspores. These 4 megaspores, in that 4 megaspores, the uppermost 3 are degenerated and the lower one is functionable one. That lower one is developed into an embryo sac. That embryo sac is nothing but here. Instead of that well-developed formation of embryo sac, there is two archegonia are formed in the place of functional megaspore. The functional megaspore is haploid in nature. And thus that functional megaspore which gives us two archegonia. Here, one important thing we have to remember, here it is a Pinus is a gymnosperm. There is a chances of formation of 2 to 18 number archegonia in each ovule. Not only in Pinus, in gymnosperm it is a character. 2 to 8 archegonia if they are present. So there is a chances of formation of 2 to 8 embryos. So it is a very familiar character, and that is abnormal character. Formation of more than one embryo is known as polyembryonic. So here coming to this archegonia, how they are developed. The development of archegonia in Pinus is similar to the gypo. 2 to 4 cells of the female gametophyte. This is the female gametophyte. This female gametophyte is developed from the functional haploid megaspore. So here 2 to 4 cells of the female gametophyte at the microbile and enlarges in size and have a dense cytoplasm and a prominent nucleus. These cells function as archegonial kinetials. Each archegonial kinetial divides periclinally to form an outer small primary neck kinetial. If we see this archegonia, here there is a small kinetial cell is present, that is called operculum, and this nucleus which is very prominent in the archegonia, that is the egg nucleus. So the primary neck kinetial divides by two vertical walls at right angles to each other, forming a neck of 2 to 4 cells. So here development of archegonia is not required according to our syllabus, even though this is one archegonium. Each archegonium consists of one opening operculum cells and 4 cell neck and one ventral canal cell and one egg cell. These are all degenerated. Only the egg cell is becoming very prominent and developed. This complete development of egg nucleus will takes the nourishment from the surrounding male gametophyte. So coming to here, now when the egg is ready, now the pollination starts. Here pollination is anemophilous. Already we discussed it with the pollinator. The pollen grains are dispersed and remain suspended in the air for some time. At the same time, the nucellular bead, this is the part is called where the microbile starts and this empty cavity and near to the archegonium. This is beak. Beak means like a nose structure which is in the ovule. This organelle is forming a viscous sugary liquid-like substance that is known as for the attraction of the pollen grains. The wind which air carries the pollen grains reach to this nucellular bead near to the ovule, and this fluid contains sugary sticky substances. So the pollen grains are coiled and stick to the sugary substance, and they slowly reach to the pollen chamber or archegonial chamber. So the pollination may be at 3-celled stage or at the male gametophyte stage. Anyhow, they reach to the archegonium. Now after certain period, each pollen tube which carries 2 male gametes, one is dropped into the one archegonium and the second one is dropped into the another archegonium. So there is a chances of number of archegonia in each ovule is 2 to 18 number. Like that, several pollen grains are reached to the ovule. So pollination may be takes at a time, and many embryos are formed. First, the one male gamete unites with the egg nucleus, zygote is formed, and the another zygote is formed in the another archegonia. So like that, several zygotes are formed, they lead to the development of embryo. So this is a very, very important common phenomena which occur in the gymnosperms, is known as formation of more than one embryo is polyembryonic. So this is the ovule, very, very important. And Pinus Reprodction is the common thing which is species available in our India. It is the mature ovule which is showing the archegonia. Now The union of male gamete with the egg nucleus of the archegonium is called fertilization. Takes place after 1 year of the pollination. So, according to the situations or according to the environmental conditions, the fertilization takes 1 year after the pollination. The pollen tube enters the tip of the archegonium by forcing itself between the cells of the nucellus. The pollen tube wall is disintegrated by the enzymes secreted from the egg, and eventually two male nuclei are released. One male nuclei unite with the egg and zygote is formed, like that embryo is formed. So pro-embryo. Now the later on pro-embryo development. The zygote nucleus divides by two mitotic divisions, forming four nuclei which move to the base of the zygote. All the four nuclei are arranged in one plane and only two nuclei are thus visible in lateral view. A synchronous division gives rise to the 8 nuclei arrangement in two ties and 2 to 4, two ties of each. In each tile, 4 cells are present. Thus, the upper group of 4 cells are formed the primary upper tile, and the lower group of 4 cells form the primary embryonic tile. So, the internal division in both the tiles forms 4 tiles of 4 cells in each. So, totally 16 cells are formed, and the pro-embryo thus consists of 16 cells. The lowest tile is known as embryonal tile, and which further divide to form an embryo. The next tile called suspensor tile elongates considerably to form the embryonal suspensor. The third tile is known as dysfunctional suspensor, and which shows abortive meristematic activity. The uppermost of the fourth tile is called upper tile or nutritive tile, which provides nutrients. So, like that, embryo will be elongate and it is developed. Now, coming to the embryogeny. The developing embryonal cells are deeply embedded into the gametophyte by the sevenfold elongation of embryonal suspensor. Thus, the several embryonal suspensors are formed. Proximal cells of the embryonal mass elongates unequal to form embryonal tubes. The cells of embryonal type are separated from each other at the time of embryonal suspensor elongation. Thus, 4 independent embryos are formed. This phenomena is known as polyembryony because more than one embryo is formed. As the polyembryony occurs due to the splitting of a zygote, it is called cleavage type of polyembryony, and only a single deep-seated poroembryo develops into an embryo, and the growth of the other embryos is arrested at different stages of the development. The proembryo divides transversely to form 2 cells, which further repeated divisions form an embryo. The embryo is comprised of 3 to 18 cotyledons. A distinct epicotyle root is root axis and a hypocotyle shoot axis with the remnant suspensors. Both the 2 years and 3 years reproductive cycles are exemplified by the Pinus species. In 2-year type, the pollination and fertilization takes place in late spring of first and second year, respectively. Third year type, the pollination takes place in spring of first year and fertilization in the spring of third year. A lapse of 2 years, the seeds shed in autumn. So now the spinous seed is endowed with a well-developed wing which is thin and papery and is easily detachable at maturity. The outer fleshy coat is called testa, and it is integument and part of oviparous scale contribute to the wing formation. Now, this is the seed. So, like that, the seeds are consist of endosperm, and the seeds are germinated in the suitable variable conditions and for the formation of new genera. So, the embryo is embedded within the endosperm, and the outer fleshy layer of integument, a part of oviparous scale, contribute to the wing formation, and the seeds are usually dispersed by the wind. So, this is about the poly